I'll get started right away. I will talk to you a bit about what we do in terms of research in my group, and I will basically give you an introduction, and the star of the show will be Ayman, who will present really what we are, one of the multiple aspects that we are pushing around doing atom probe tomography on liquids. Just as a bit of background, materials that we use every day, that we see, that on which I'm sitting right now, or that you're using to watch me or listen to me, they're all around us, but they draw their properties of interest from a very complex structure and microstructure of starting from just a few atoms getting arranged on a crystal lattice. And then if you go on a slightly bigger scale, you have all of these individual crystals of matter that meet, and you have this intersection between different grains, different crystals, different that we call grain boundaries, and there's different elements making these different materials that also all are distributed according to the physics and chemistry of the material. And when you start looking at this architecture from just an atomic size up to millimeters or meters, then this all plays together to provide the materials, the properties that we are interested in. So it's quite obvious that one technique cannot answer all of the questions of how the atoms are arranged up to a matter. So we typically use a range of different analysis techniques, using often a source of light, for example, with X-rays or optical microscopy. We're using visible light, or we can use electrons, for example, that we send onto the sample, and then we look at what comes out or what goes through. And we can also use ions, so ions that are accelerated onto the sample, and then we look at what comes off. And the technique that I'm going to really focus on today, laser pointer here, is called atom probe tomography. And it's quite interesting in terms of the statistics, the sensitivity that we can reach with atom probe. We can go down to measuring small amounts of matter, and on very small, on a small scale, almost down to individual atoms. And a lot of the techniques that are more often encountered, like scanning electron microscopy or transmission electron microscopy, are good at looking at the way atoms are arranged, but not always, so the structure of the matter, but not always in which atom is sitting where inside of the structure. This is especially true for light elements, so typically carbon or boron or hydrogen. And atom probe is actually good at looking at these things. So instead of trying to solve all of the problems of the material world with just one technique, what we aim to do increasingly in my group and more broadly at the Institute and more broadly in the scientific community is to combine information from the different techniques. So if I look at, for example, this precipitate strengthened steel, austenitic steel, you can see that there's here a structure with inside some contrast, and that contrast corresponds to particles, precipitates that are formed inside of the matrix. And these precipitates contain manganese and aluminum and a bit of iron and carbon. And if I look at really high resolution transmission electron microscopy, I can see how the atoms are arranged in these different parts of the material. So here you have what we call a channel that is completely disordered with iron, manganese, aluminum, all distributed randomly. And then inside of the precipitates, these atoms are arranged on in a very specific way alongside the carbon. The problem is that these techniques like TEM typically give us just one projection through the structure. We don't have the full three-dimensional aspect of the matter, but also, for example, we don't really know how to measure the content of carbon inside of these particles. So this specimen that I'm showing here has a weird shape, and I will get back to this afterwards for why, but this is a specimen that then we can put into the atom probe. And in the atom probe, we get a full three-dimensional compositional field. So we can measure how much of carbon is inside of some of these precipitates that we see, but also how much aluminum, how much manganese, how much iron. And all of this information is useful to understand how we can form these materials in a specific way, or how we can improve their design to get better properties, for example, make them stronger or more resistant to corrosion, or depending on the application that we have in mind. So now I'm going to step by step, and I'm not going to quote his 17 here, but step by step, I'm going to take you through how we do atom probe. So the principle of atom probe is actually pretty simple. If we take a surface that is made of atoms like this, and we consider one atom on the surface, and we start applying a high field, a high electric field. So basically we are putting a lot of positive charges on the surface by sucking in the electrons by applying a high voltage. Then what happens is that surface atom starts to be repelled from the surface and turn into ions. So basically this atom has been pushed away and lost 1, 2 electrons. When you have an ion inside of an electrostatic field, the electrostatic field will contribute to accelerating this atom, this ion, sorry, and it will project it away from the surface. For this to happen, this process that we call field evaporation, we need fields that are in the range of 10 billion volts per meter, so 1 to 6, 10 to the 10 volts per meter, which is enormous. So how can we reach such fields? We can reach a very high field by looking back at some electrostatics that you might have done during your undergraduate. But if you take a sphere of radius r and you charge it to a voltage V, the electrostatic field at the surface of the sphere would be V divided by R. What we can easily make is a sharp needle, and the end of this needle has almost a spherical cap. So what we're going to have is a field that is almost V divided by R. There's a small constant here that is typically 3 to 8, depending on the specimen shape. But it means that if I apply a few kilovolts on a specimen that has size in the range of a few tens of nanometers, then I can fairly easily reach the field that I need to do atom probe, or to remove the atoms from the surface. So this is exactly what we do. We take a piece of material and we shape it as a sharp needle with a radius of curvature that is about 100 nanometers. So it's like maybe 100 to 1,000 times smaller than one of the hair that you may have the chance to have on your head. I don't have many of those anymore, but that's the idea. And we cool this specimen down to cryogenic temperature, so between maybe 25 Kelvin, so minus 250, and 80 Kelvin. And we place this specimen in front of an electrode, because the strength of the electrostatic field is always the voltage that we apply to the specimen and wherever is the next ground. So we put this about 40 microns away to increase the strength of the field right here. And we apply this voltage, a few kilovolts, up to the point where the surface atoms are going to turn into ions and be projected away. The field is not just at the surface. The field, the electrostatic field, is all around the specimen, and it distributes in a way that the ions that are formed are going to fly away from the needle apex and fly and fly and fly and fly and fly. And then we just collect them on a particle detector. So it's a specific type of detector that can grab one ion and record its position, where it landed on the detector. And what we can do afterwards is either use a DC voltage and high voltage pulses to temporarily increase the field, or we can also use short laser pulses that will last a few tens of picoseconds, or about 10 picoseconds, to increase the temperature during a short amount of time. And this will trigger the time when the atoms actually turn into ions and start their flight. So what we can do, if we know when the ion starts its flight, we can also, when it lands on the detector, we can record this time of flight. And the time of flight is proportional to the mass of every single one of these ions. So hydrogen, which is the lightest one, will travel very quickly from the specimen to the detector. And if we take something like tungsten or uranium, which is somewhere down the periodic table, it's really heavy, it will take a long time to reach the detector. But it means that I can take my specimen and then peel it off atom by atom. I'm destroying my specimen atom by atom. And I can record the position and time of flight of every single one of these ions that I've removed. And afterwards, using a bit of magic, I can turn this information of time of flight and impact position into a three-dimensional map of where the atoms were sitting inside of the material before we removed them. There's no magic. I will tell you how we do it. So the two aspects of the results that we get is first what we call a mass spectrum. So we peel the ions, we measure their time of flight. We can turn this into a measurement of the mass of each ion. And we can put together a histogram of how many ions we collected that has a specific mass to charge ratio. So here, if I zoom in, I can see that I can detect the chromium here that has multiple isotopes, the vanadium that has mostly one isotope, and then the 4 isotopes of iron, and then the 1 isotope of manganese. So I can go down to measuring the individual isotopes of a specific element. But globally, what this gives me is the relative quantity of, for example, iron versus chromium versus manganese versus vanadium inside of this material. So I have the composition of the material. And what I also have are these detector coordinates, the impact position, and I can turn them into a series of X, Y, Z, and mass information in the three-dimensional space. So the way we do this is that we assume that the atoms are sitting on the spherical cap at the apex of the needle, and they reach the detector. So they follow a certain projection from the surface onto the detector. And so we assume that we have a clue what this projection law actually is, and we can backtrack this impact position onto a virtual emitting surface. And every time we've processed this ion and position it back onto the surface, we move the surface down by just a tiny amount. And this tiny amount is supposed to reflect the way the atom contributes to the overall volume of the specimen that we've imaged. And then we do this for a second ion, and then for a third ion, and so on and so forth, sequentially. So the data is not perfect. The projection that we have is not perfect, but it's good enough. So because of the distribution of the electric field near the specimen surface, which is actually not completely straightforward, we have small trajectory aberrations that we cannot really control. And the particle detector that we use misses some of the atoms, but it's not related to their mass to charge or their mass. So we can detect heavy and light elements. It's random on the detector what we lose. So when we start with a specimen that's really well ordered with atoms on atomic positions, at the end we have a bit more of a mishmash, but with a spatial resolution that is better than half a nanometer typically. So it's not too bad. And we have the composition. We have locally on a sub-nanometer scale, we can measure how much atoms of each species we have, which is quite unique and all in 3 dimensions. No other technique can offer this. So again, if I look at the piece of steel that we analyzed that corresponds to this mass spectrum that I showed you before, we have this point cloud. Every point here is 1 ion that was detected, reconstructed. And the strength of the atom probe is in these different maps. So I can see, for example, where is the manganese in this microstructure? It's in this big particle that also contains carbon because it's a carbide. And this carbide is actually sitting at a grain boundary. And we have some defects here, dislocations that are bearing some vanadium that is segregated. And all of this information then we can use to try and make materials better or understand better how they deform. And this is really the purpose of what we do in the Institute, as Professor Haber was saying in the video. So when I was a young student, a long time ago, maybe 15 years ago, there were about 20 atom probes in the world, and now there's probably about 100. So there's quite a density in the United States, and we have quite a high density also in Europe, but MPA is the best equipped lab. We have 3 machines and 3 focused ion beams that are the microscopes we use to make the specimens. And my group is quite large. I mean, it's about between 20 and 25 people, depending on how we count and on the phase of the moon, I guess. And these instruments are complemented with all of the other fancy equipment that we have. And you will see some of these presented in the coming talks over the next few weeks. So what we do in my group is always to use these advanced microscopy techniques on these materials that have interesting features at the nanoscale, and we try to bridge some of this with simulations, especially with our colleagues from the computational department, some of whom are going to also present in this seminar series. And so we work, for example, on engineering alloys that go into the plane engines, for example, or that will go into cars, or that might actually also find their way into hip implants or that types of things. We have a fairly large activity on hydrogen, trying to understand how hydrogen goes inside the materials, what it does inside of the material, and how it causes mayhem and causes materials to break down. And then we're also trying to push on the application for functional materials, so typically catalysts or magnets or thermoelectrics. And everything we do is in the context of trying to make materials, understand better how materials behave, to try and make them better in the end. So if I look at a turbine blade, for example, we can go down to looking at the tens of nanometer scale, and these are strengthened by these gamma prime precipitates, for example, inside of a gamma matrix. So the matrix again here is disordered. The precipitates are ordered, so atoms of specific species are sitting on specific sites inside of the crystalline lattice. And then in the atom probe, we can look at what is sitting where, which atom is sitting inside of the structure, in the particles or in the matrix. I don't know why this is green, but this is turning all green. Anyway, so this is this full 3-dimensional information, and then we can combine this with transmission electron microscopy to look at the way the materials deform and the way the atoms are rearranging because of the deformation. So this is work from Paraskev-Askontis, for example, looked at some carbides in these types of alloys that go in high temperature turbines to try and understand how the structure of the material is changing during service and what causes it to break down, basically. We also have an activity on materials that can be used to split water, for example. So these are really advanced electron microscopy techniques where we can look in 3 dimensions at what the materials look like. There will be also a talk from Tina Choi in this series on similar types of materials. And we can see that, for example, the material, after it's been partially reduced, so we've removed a bit of oxygen from the sample, it makes it a much better catalyst for splitting water, so getting hydrogen and oxygen out of water. And when we do the atom probe on these types of samples, we can see that there's basically segregation of impurities, boron, sodium, and nitrogen from the fabrication process, sitting at the surface and stabilizing the oxygen vacancy, so the oxygen that is missing on the surface. And this explains why this material, after partial reduction, behaves much better, is much more active catalyst. So what we tend to do in my group is try to really push the frontiers in atom probe research. So here is where I'm going to start getting into hydrogen, and I'm going to lead to Ayman's talk. So as I've mentioned before, hydrogen is interesting as a scientific problem, but it has a lot of engineering applications. So if you take a nickel-based superalloy again and you pull on a small piece of material until it breaks, you can deform it about 45% before, and you have to pull very hard before it will actually break. And if you put hydrogen in and do the same test, instead of deforming it 45%, you will only deform it 10% before it breaks down. So there's this phenomenon called hydrogen embrittlement, and hydrogen is the smallest and lightest element. It can penetrate inside of the material and interact in all sorts of weird ways with defects, with grain boundaries, for example, with the surface. And there are many, many mechanisms that are at play here, and we don't necessarily know which one is the most active or which one is activating this behavior. So in this, we don't know because we can't really measure where hydrogen is in the structure. And atom probe is actually quite good at this in principle. It's not without challenges, but in principle, we can do it. One of the challenge, for example, is that an atom probe is done in a UHV chamber. So there's always a bit of hydrogen inside of the chamber that is going to affect our measurements. But also when we are preparing our specimens, we worked out that very often, and during the preparation of these needles, we introduce some hydrogen from the other microscopes that we use to make the sample. So we've managed to put together a suite of microscopes. As I said, we can't do everything with one machine. We need to combine them all together. And we are using this combination of an atom probe and a FIB to make the needle. And we can do this at low temperature, at about minus 200 degrees. And we transfer specimens from one machine to another with these UHV suitcases. So a small vacuum chamber that is also cooled down to very low temperature. And if we do this properly, then we avoid this introduction of hydrogen. And it means that when we do the atom probe, we actually have a very low level, background level of hydrogen. So this is a titanium alloy that is developed to go into plane engines as well. So it's actually quite important to understand when hydrogen goes in, what does it do to the sample? Because when you start cycling up and down in temperature, when the plane, for example, takes off or lands, we need to be able to understand how hydrogen is going to behave in there, because at some stage it might start to create hydrides, and these hydrides will be brittle and cause failure, i.e. the blade is going to break and the plane engine is going to stop working, which is really not a good thing. So we are, for example, working on putting hydrogen inside of the sample and looking at the way it at high temperature, in that case a few hundred degrees, and looking at the way the elements from the alloy are actually changing their distribution in 3 dimensions at the few nanometer scale. And we can see the formation of intermetallic phases and all sorts of hydrides that were not necessarily predicted from thermodynamics. And the next step in this is using the same setup to look at liquids. And for this, I'm going to hand over to Ayman El-Zoka to give his presentation.
Okay, many thanks, Baptiste. Hi, everyone. It's a pleasure having the opportunity to share our latest results. Can you see my screen?
Yes.
Great. So I'm just gonna take the next few slides as an opportunity to show our recent advancements in the field of doing atom probe tomography research on liquids. As Baptiste have beautifully explained, we're always trying to push the barriers a bit into analysis of materials using atom probe tomography, because we believe it can bring on a lot of insights. So here it goes. We wanted to do atom probe tomography on water, and I'm gonna provide the reasons in the next slide. But if we wanna do so, what do we need to make it happen? We need electrical conductivity, PAS. The water is an insulator, and there is very little scarce data on its conductivity, especially when solid. We need to be able to make a tip out of it, and we need to be able to make a tip out of it. We need to be able to shape it using the fibbing techniques that we have. We need it to be placed onto some sort of a substrate, a suitable support where there is sufficient adhesion for it to go through this atom probe tomography analysis without breaking off or without catastrophic failures. The radius of the tip needs to be within 50 to 100 nanometers, and it needs to clear off against its surroundings with at least 2 to 3 microns. And we did try conventional approaches by using silicon post coupon. On each of these silicon posts, if you have a metal and you want to analyze it, you would put the tip of metal or alloy here. We wanted to do that with water, excuse me. So We just basically tried to put a drop in there, freeze it, and then take it to the PFIB using the cryo suitcase and the LAPLACE project equipment that Baptiste have just shown. But when we try to do so, we find out that the droplet of water, as you can see here, is really big. It's really big, and it's really hard trying to mill away all of this ice, mainly because the silicon is very hydrophobic, so it doesn't spread all over, and also you don't know where it goes. So it's very non-controllable, and even if you can make a tip out of it, there is very little access to the tip with the laser because of the clearance issue. So we wanted to try and make a protocol for this to happen. And my previous work in the University of Toronto during my PhD came into play, where I looked at the de-alloying of silver gold alloys to make a sponge out of gold after dissolving the silver in acidic conditions. And then I tried to look at these sponges using atom probe tomography. So we had to fill in the voids with copper electrochemically. Long story short, we needed to fill it with something. So what if we can just fill one of those sponges with water, and at the same time we can maybe provide more conductivity to the water structure while looking at the and at the same time try to have more specific site-specific access to the water once it penetrates this nanoporous gold. So we tried to use nanoporous gold as a frame for analyzing water with atom probe tomography. How could this be impactful? Obviously, what was moving us in this aspect was not only being able to analyze the frame itself, the nanoporous gold, which is opens the door to a lot of corrosion analyses. We could also embed things in the solution itself. We can look at solvated ions, we can look at nanoparticles, we can look at biological structures, and so on. So making this work, we could do, it would open the door to a lot of new exciting things. So we went ahead and tried it out. We would basically de-alloy silver gold, just like I did in my PhD, in nitric acid. Then I put it in water. This is not supposed to be nitric acid, it's supposed to be D2O, deuterated water, so that we can differentiate between the hydrogen coming from the solution that we dipped it in and the hydrogen coming from the analysis chamber, as Baptiste have explained. Then we take the clip after immersing it in D2O or NaCl D2O, and we quench it in the glove box to eliminate moisture using liquid nitrogen. Then we use the CRYO-TRANSFER suitcase to take the sample to the cryo-PFIB, where we would do the prep to make tips out of it. And I'll show in the next slide how so. And then we take it to the 5000 XS atom probe tomography machine, where we can analyze those tips. So we did actually go with this crazy plan, which we didn't know it was going to work. As you can see here, this is an SEM image of the surface of the nanoporous gold and the ice covering most of it. So we went onto the edge and we kind of chose this area where we could work on our, suspected tip by sharpening it further and further using plasma FIB, which is basically a beam of plasma. And then we kept trying to sharpen it, and we were pushing the limits. We didn't know what we were getting at all. And then we arrived at this shape. You can see here that there is a little nano icicle on top of the nanoporous gold post. If you're doing conventional atom probe tomography, you would get rid of this icicle. But that's what we were looking for. And there is no frame in this case. That's what we ended up with, this nano icicle of water. And we knew that, it could be non-conductive, but we actually wanted to try it out anyways. And we got pretty exciting results, mainly that it didn't break. We got a lot of data out of this tip and other tips as well, where we could actually analyze water on the nanoscale and the atomic scale. We detected clusters of D2O at different, with different degrees of polymerization of these D2O molecules, going from one cluster to fourth and even say up to the sixth cluster. So we could say a lot about the field evaporation of water for the first time, and also about possible mechanisms on why this is, how come this seemingly non-conductive solid is actually conductive at the end. And then we went down further through the tip, and we were actually able to get into the nanoporous gold part, where we could beautifully see the nanoligaments, the gold and the silver. Interpenetrated between them is the water with the NaCl concentrations. We could analyze the water inside the sponge, and we could analyze how it interacts with the solid once we froze it. So it's pretty exciting, and it shows that we can take a look at the liquid-solid interface more closely, and that opens the door towards that area. This is just to show the different spectra that we were able to get with chlorine and with sodium, because we were really interested in knowing if we can detect solutes inside these nanopores. We were also able to detect little floating silver that is from the de-alloying procedure, well above the nanoporous gold part. And that was, again, an unprecedented observation. And it would only be enabled through this cryo-APT procedure, where you can catch the de-alloying in situ. You don't have to dry it out. You don't have to take it out of the medium and put it into a copper plating solution. You can just use water and directly freeze it and take it to the PFIB. Why was this possible? We're still looking at explanations, but we give a few nice postulates in our recent paper in Science Advances, titled Enabling Near Atomic Scale Analysis of Frozen Water. Please check it out if interesting. And we kind of draw onto the effects that are presented in our technique, where instead of just depositing a layer of ice on top of a metallic tip, and that was the conventional approach with these liquids, where there was seemingly less conductive ice because of the different concentration of the electric field being concentrated instead of at the tip of the ice. It was concentrated at the metallic tip. However, in our case, we think because we had a full tip made out of water that is anchored by these nanopores, we were able to get a concentration of the field at the ice vacuum interface. So that kind of enabled the prolonged and the consistent field evaporation of these water molecules and or polymers. So yeah, it's pretty exciting, and we're trying to look into different things to do with this right now, understanding the phenomena more, and at the same time trying to look at applications in nanomaterials, corrosion, and even biological structures. So just at the end, I would like to conclude the presentation by stressing that atom probe tomography is able to provide 3D compositional mapping with sub-nanometer resolution in bulk samples, and of various natures, metallic, thin films, oxides, etc. We can also see through this presentation that atom probe tomography is pretty cool, and it overcomes a lot of, it gives a lot of advancements into characterization of materials, but it has a lot of underlying physics that is not easy to understand, and there is still a lot of work onto that. Finally, we can also see that atom probe tomography gives these insights that you cannot get with other techniques like TEM, like XPS, and like SIMS. So, yep, the world needs more atom probes. And if I will conclude this by thanking Baptiste first for letting me join, and thanking Sophia and the people at the RCO for organizing this.